Researchers at Tufts University have made a groundbreaking leap in the field of regenerative medicine with the creation of Anthrobots, small biological robots constructed from human tracheal cells. These multicellular robots, ranging from the width of a human hair to the tip of a sharpened pencil, self-assemble and have exhibited remarkable healing properties, encouraging the growth of neurons in damaged regions within lab dishes. The Anthrobots, described as a potential new therapeutic tool for regeneration and disease treatment, are an evolution of earlier research that produced Cenobots from frog embryo cells. What sets Anthrobots apart is their derivation from adult human cells without genetic modification, opening the door for personalized therapeutic applications. In a study published in Advanced Science, lead researcher Michael Levin and PhD student Gizem Gumuskaya revealed that Anthrobots not only create new multicellular shapes, but also exhibit unique movement patterns. Their capabilities include propelling across surfaces and stimulating neuron growth, particularly in damaged areas. The Anthrobots, which range in size and movement patterns, can last for a few weeks before naturally breaking down. Their unique properties make them ideal candidates for therapeutic applications, as they can be constructed from a patient's cells without triggering an immune response. Furthermore, Anthrobots can be easily reabsorbed into the body after completing their tasks. The study demonstrates that Anthrobots, derived from tracheal cells that typically lead a quiet existence, can be reprogrammed to create new structures and perform diverse tasks. This ability to manipulate cell interactions opens avenues for designing multicellular structures with specific functions, analogous to arranging building elements into different structural forms. The researchers observed that Anthrobots, when arranged in a clustered assembly known as a superbot, could encourage neuron growth across damaged regions. Although the exact mechanisms behind this phenomenon are yet to be fully understood, the study showcases the unexpected capabilities of Anthrobots in promoting healing. The advantages of using human cells in Anthrobot construction include scalability, with the ability to produce swarms of these bots in parallel. Additionally, Anthrobots exhibit no risk of unintended spread outside the lab, do not reproduce, and involve no genetic edits, ensuring safety and predictability. As Anthrobots move beyond the confines of laboratory conditions, their potential applications extend to various therapeutic scenarios. The researchers envision Anthrobots contributing to tasks such as clearing plaque buildup, repairing nerve damage, recognizing abnormal cells, and targeted drug delivery. The study marks a significant step forward in understanding cellular assembly, opening new avenues for regenerative medicine and the creation of innovative biological structures. Thanks for watching. Please see the citation in the description. If you want to see more research video like this, please like, share, and subscribe our channel.